Yes, students. Now let us start our next topic, which is types of germination. Now, before studying types of germination, you need to study two more terms related to the germination. See, you have studied now the structure of the seed, bean seed, and you all know that these are the cotyledons. This is the plumule, we have read, and this is the radicle, which is going to form the root. Now see, these two cotyledons, they are attached with the embryo at a point. This point is known as embryo axis. Embryo axis. The point where the two cotyledons are attached with the embryo. Now you must see carefully that the point between the plumule and the embryo axis, this much part, the part between the plumule and the embryo axis is known as epicotyle. The part between the embryo and the plumule is known as epicotyle. And the part between the embryo axis and the radicle, this much part, this will be known as hypocotyle. Hypocotyle. Now during germination, during the seed germination, both epicotyle and hypocotyle will never elongate together. Either the epicotyle emerges first or the hypocotyle emerges first. So depending upon this, the emergence of the epicotyle or hypocotyle, the germination is of two types, epigeal and hypogeal. See, you can refer your book. The region of the axis between the point of attachment of cotyledons and plumule, the distance, that is called epicotyle, and the region of the axis below the cotyledons is called hypocotyle. Both epicotyle and hypocotyle of seed never elongate together during germination. It is either the epicotyle or hypocotyle that elongates. If epicotyle elongates, the cotyledons remain underground. See, you can see the germination of pea plant. Here they have shown, this is the seed. Okay, now we have sown the seed in the soil, suppose. Now the radicle starts emerging out. If the radicle is coming out, means this from here the radicle starts coming out, and from here the plumule. Now this epicotyle starts emerging. Epicotyle, the region between the embryo and the plumule. This region, this starts first. So when it emerges, slowly and slowly it will elongate. Then the cotyledons will remain under the soil. You can see here the cotyledons remain below the soil, and epicotyle elongates and it forms the shoot. Plumule comes out, it forms the shoot. Such type of germination where the cotyledons, they remain below the ground. Cotyledons remain below the ground. This type of germination is known as hypogeal. The cotyledon remain underground and germination is called hypogeal. Hypo means below earth. Geo means earth, below the earth. Okay. Now on the other hand, you see bean seed. In bean seed, this is the bean seed. What happens that the hypocotyle comes out first. Hypocotyle. When the hypocotyle comes out, see, this hypocotyle will come out first and it pushes the cotyledons. It forms a sort of arch. This is the loop or the arch. And as the hypocotyle will come out, the cotyledons are also forced. This is trying to come out. It forms a sort of loop or arch, and then it becomes straight. See, it becomes straight, and both the cotyledons, they also emerge out. And the cotyledons come above the ground, and then they form the rudimentary leaves, and then the other leaves will come out. So this type of germination where the cotyledons come above the ground is known as epigeal germination. Epigeal means above the ground. So in pea plant, in some plants the germination is hypogeal, in some plants the germination is epigeal. It depends. But what is the difference? The difference is that if the cotyledons come above the ground, above the soil, the germination is called epigeal, and if they remain inside the soil, do not come out, then it is called hypogeal. So you can see here Note the cotyledons remain underground, and this kind of germination is called hypogeal. So this was in P, and this example they have given of bean seed. In bean seed, the germination is epigeal. So another they have shown is maize grain, germination in maize grain. In maize grain also, you know that radicle is protected by coleoptile and coleorhiza, and plumule is protected by coleoptile. So the radicle emerges out and plumule also emerges out after breaking the covering, the sheaths, coleorhiza and coleoptile, and then this radicle, it soon goes off. It is means it is lost, and then it is taken up. This root will be broken down, and then the next roots come out. We call them adventitious roots or fibrous roots. So here the plumule pierces through its protective sheath, coleoptile, and grows straight upwards. The plumule grows upwards and it can be seen as membranous covering. The cotyledon, which was the scutellum, absorbs food from endosperm till it is exhausted. So the cotyledon has remained inside only, it will not come out. So this again we will say the germination is hypogeal. So there are two types of germination. The difference they have given, and you need to read it again. Hypogeal germination, cotyledon remains underground. Epigeal germination, cotyledons pushed above the ground. Here epicotyle elongates faster, and here hypocotyle elongates faster. This is usually in monocotyledon seeds, and epigeal germination occurs usually in dicotyledonous seeds. Then in some plants, in some special plants, there is viviparous germination. Viviparous means this is a special type, and this occurs in mangrove plants. Now you would like to know what are mangrove plants. These are the specialized plants which grow in marshy areas. Marshy areas means like coastal areas where the soil is rich in lot of nutrients. The muddy soil or the coastal areas, near the coastal areas, we call them mangrove plants. The examples are Rhizophora and Soneraceae. The two examples they have given, they have some special type of germination which does not resemble any of these. So in these type of plants, what happens that seeds germinate inside the fruit while it is still attached to the parent plant. The parent plant, the fruit is still attached. Look at this plant in Rhizophora, viviparous germination. This is the fruit and this is the main plant, parent plant, and it is still attached, and while being attached the radicle has started coming out, hypocotyle radicle. Okay, then after the plant grows, then it is later on detached, and then it will fix itself in the mud, and then it will form the further leaves. So this type of germination is called viviparous germination. Okay, your last topic is the seedling. So he says that germination here ends when the seedling has come out. That is the end of the germination. Now seedling is an independent plant. Now the new leaves, first the green stem is also green, so stem also photosynthesizes, as well as the new leaves which come out. Now they are capable of performing photosynthesis, as well as the roots which have come out, they can absorb nutrients from the soil. So this was all about your chapter. Now let us revise the progress check. Here we have some fill in the blanks. In a dry seed, the embryo is in dry seed, embryo will be quiescent. Please tick mark quiescent. Quiescent means inactive or dormant. Water is absorbed by the seed mainly through micropyle. Very low temperature dash the growth of embryo. Very low will inhibit. Very low temperature will inhibit the growth. Germination in P is, in P, just now you have read, it is hypogeal. Underground, the cotyledons remain underground, so hypogeal. Coleoptile is a part found in the germinating maize. Alkaline pyrogallic acid is used for absorbing oxygen.